Welcome all to the video lecture on Louis Pasteur and his contributions to science. I am Dr. Dhanya from the Department of Microbiology, St.Mary's College, Thrissur. As we all know, Pasteur is known as the father of immunology and microbiology. He is a biologist, microbiologist and chemist. He was born in Dole, France in 1822. His father was Jean-Joseph Pasteur, who was a tanner and a surgeon major who received the Legion of Honor during the wars. Pasteur earned his bachelor degree in arts and in science from the Royal College of Besançon, and he received his doctorate from the École Normale in France. In 1848, he became a professor of chemistry at the University of Strasbourg. Coming to the contributions of Pasteur in the field of science, in 1849, Pasteur observed tartaric acid and racemic acid mixtures, and there was a controversy on these two molecules. Even though the molecules were having the same chemical structure, their chemical properties were highly different. Pasteur solved this problem by his intense inspection using a microscope, during which he identified that these molecules were identical, but they were having different spatial arrangements. These molecules were mirror images of the other. This discovery has led to the branch of science, stereochemistry. His next work was to disprove the spontaneous generation theory, which was a theory which was there until the second half of the 19th century. Many scientists and philosophers believed that there is a spontaneous generation, that is, life could arise spontaneously from non-living matter. Pasteur disproved this theory by his experiment on swan neck flask. In this experiment, he demonstrated that sterilized and sealed flask containing a culture media could form no growth, while the sterilized growth in open flask will develop microbial growth. He suggested that microorganisms from air contaminate sterile solutions, but air itself does not create microorganisms. His next study as a professor of chemistry and dean at the University of Lille during 1854 to 1862 was on fermentations. He found solutions to the problems associated with alcoholic drink manufacture. His studies showed that living cells or yeast are responsible for forming alcohol from sugar, and also he showed that contaminating microorganisms are the causative agents which could cause the fermentation to go sour. In 1863, at the request of the Emperor of France, Pasteur studied wine contamination and showed that the contamination is caused by the growth of microorganisms. And he devised a method to prevent the contamination. He showed that by heating wine to up to 50 to 60 degrees Celsius, the wine could be stored for longer times. This process is now known as pasteurization, and this is used for the preservation of many foods and beverages even now. In 1865, Pasteur rescued the silk industry. During that time, there was a disease known as pebrine or pepper disease. This is a disease of silkworms caused by a protozoal parasite known as Nosema bombyces. The silkworm larvae having this particular disease were observed to be covered in brown dots, and they were unable to spin silk thread. Pasteur identified the cause and developed a method known as graining, through which the elimination of contaminated females were possible for the preservation of healthy eggs, and this technique was adopted by silk producers throughout the world. His next contribution is the discovery of Pasteur effect. Pasteur identified that the fermentation process could be arrested by passing oxygen through the fermenting fluid, and this is known as the Pasteur effect. He concluded that this is due to the inability of the microorganisms to carry out fermentation in the presence of oxygen, and he introduced the terms aerobic and anaerobic to designate organisms that live in the presence or absence of oxygen. In 1879, Pasteur was working with the disease fowl cholera or chicken cholera, which was a very serious problem in chicken coops at the time. He observed that the bacteria which is responsible for this particular disease, Pasteurella multocida, when inoculated into a chicken, the bird will be killed. But when a weakened culture of the same bacteria was used to inoculate the chicken, the birds remained healthy. So, through this, the principle of vaccination with attenuated pathogens were discovered. In the same year, Pasteur worked with the disease anthrax, which was caused by a bacteria known as Bacillus anthracis. Anthrax is an infectious disease that affects cattle, sheep, etc. And this is a zoonotic infection which could be transmitted to human beings. And this disease was causing killing of large populations of sheep in France, and this was causing serious problems to the economy. Pasteur developed a mild, harmless culture of the bacteria, and he used this vaccine to vaccinate hundreds of animals. So, through this work on anthrax and in chicken cholera, Pasteur demonstrated that vaccination or immunization is possible by making the causative agents weakened through artificial methods to make attenuated vaccines. Pasteur gave these organisms the name vaccines in honor of Jenner. In 1882, Pasteur worked with an epizootic of swine erysipelas, and he was able to develop a vaccine against this particular disease. He also worked with several other bacterial infections such as the infections caused by Staphylococcus, Streptococcus, Pneumococcus, etc. His next work was on the disease rabies, which is a highly contagious infectious disease. It is a viral disease, and it was transmitted by the bite of rabid animals. In 1885, Pasteur made a weakened virus suspension of rabies and used this to inoculate a boy named Joseph Meister, who was bitten by a rabid dog. And through this vaccine administration, Pasteur was able to rescue this particular boy. To help to treat future cases of rabies, Pasteur, with monetary donations coming from all over the world, instituted Pasteur Institute in 1888. So, his medical discoveries proved germ theory of disease. Even though he was not the first to propose the germ theory, his experiments indicated the correctness of germ theory. He is regarded as the three main founders of bacteriology, together with Ferdinand Kohn and Robert Koch. So, coming to the chronological order of Pasteur's contributions to the field of science, as a physicist and chemist, he worked with crystallography, alcoholic fermentation, and as a biologist, he disproved spontaneous generation. He devised pasteurization, detected Pasteur effect, and worked with silkworm diseases. As a microbiologist, he could develop vaccines against several diseases such as anthrax, rabies, chicken cholera, and swine erysipelas. Louis Pasteur was the director of the Pasteur Institute until his death, and his body was interred in a vault beneath the institute. Pasteur was a chemist who fundamentally changed our understanding of biology. He is remembered as one of the founders of preventive medicine. Few people have saved more lives than Louis Pasteur. The vaccines he developed have protected millions. His insight that germs cause disease revolutionized healthcare, and he found new ways to make our food safe to eat. Even after his death, he continues to inspire us through his famous quotes. To cite a few, chance favors the prepared mind. Little science takes you away from God, but more of it takes you to Him. Gentlemen, it is the microbes who will have the last word. The role of the infinitely small in nature is infinitely great. It is not the germs we need to be worried about, it is our inner terrain to be worried about. There are no such things as applied sciences, only applications of science. When I approach a child, he inspires in me two sentiments: tenderness for what he is and respect for what he may become. Science knows no country because knowledge belongs to humanity, and it is the torch which illuminates the world. Science and peace will triumph over ignorance and war. And this is the last words of Louis Pasteur: one must work, one must work. I have done what I could. With these words, let us wind up this video lecture. Thank you all.